Today I'm going to demonstrate example of application of mathematical mechanistic modeling approach to predict and analyze clinical trial results in neurodegeneration area. The main goal of quantitative systems pharmacology modeling is to provide mechanistic description of key pathophysiological pathways as well as different mechanism of action for therapeutics. In fact, unlike different empirical models or correlation analysis, such platform is capable of predicting dynamics of biomarkers. So today I'm going to describe our Alzheimer's disease QSP platform that we have been developing for about 10 years in collaboration with different pharma companies. And then I will demonstrate how we applied our platform to describe aducanumab clinical trial data and how model predictions align with the data. And finally, I will show the capability of platform to make some extrapolation from preclinical data to simulate different types of combinatorial treatment. At that stage, our Alzheimer's disease platform was focused on the description of two key biomarkers, such as amyloid beta and tau, because they are most commonly targeted in clinical trials. And we have divided the brain to three regions: entorhinal region, limbic system, and neocortical region, to be able to describe in a very simple way Bragg stages of tau pathology accumulation. So let's see what's going on and what interplay of processes we can see in neocortical region. The production of amyloid beta oligomers, which are the substrate for formation of toxic oligomers or protofibrils of A beta in intracellular space, is tightly regulated by other cell constituents such as lipids and sphingolipids. Similar aggregation model was described for tau protein because it also can oligomerize, but in addition, it can also form insoluble neurofibrillary tangles intracellularly. Important point in our model is key role of tau protein in regulation of transport, because it is microtubule stabilizing protein, and transport regulates autophagy process, which is a key process for degradation of toxic waste in the cell. This is a very simplified sketch of the model. In fact, of course, there are much more variables, processes, and interactions between them. Toxic species such as oligomers or tangles can activate other signaling processes such as p53 or, for example, caspase, and these processes may further lead to neurodegeneration. So finally, our model was able to describe Bragg stages of tau pathology, and it was also able to describe heterooligomerization of amyloid beta, because we consider different types of A beta monomers, such as A beta 40, A beta 42, or even proglutamate modified amyloid beta. So more details you can find in our publications. We have developed specific model for intraneuronal processes because we assumed that amyloid and beta pathology are tightly connected through different intracellular mechanisms of regulation. Some of them have been mentioned before on the previous slide. So moreover, we assume that some of these intracellular mechanisms can be potential pathological triggers. We considered simplified description of different modules of cell metabolic systems, which can potentially contribute to the protein degradation, as well as degradation of toxic species, to the APP, amyloid precursor processing, to the protein secretion or endocytosis. To calibrate this part of the model, we used available healthy baselines. We used a lot of data for perturbation of intracellular systems in vitro, for such as different drug effects in vitro. And finally, this model have been validated on Alzheimer's disease data. In fact, this kind of model can be a framework for better understanding the cell state based on possibly measured biomarker data in cerebrospinal fluid if we have available data on cell metabolism. This model have been also used to find optimal model of disease drivers disease progression model. Among the different types of cell processes, we have selected potential disease drivers, which could independently of A-beta or tau change with time during the time interval between 60 and 70 years. We have selected them mostly based on the data about their very early appearance at the very early Alzheimer's disease stages, or we have chosen among potential genetic risk factors such as ApoE4. We combined them. We created a lot of combinations of these potential disease drivers and performed simulations to compare with the data on different disease progression markers. Some of them are shown here. And so our model was able to describe qualitatively the age-dependent change for different biomarkers of breakdown of cell metabolism, such as lysosomes, cholesterol, ceramide, autophagosomes, and so on, as they demonstrated, for example, increase or decrease. Then we have selected the best 100 combinations based on their mean square error across this data set and calculated frequency of appearance of different disease drivers in these combinations. This allowed us to formulate more or less appropriate disease progression model. This model is very important because disease progression independent of A-beta and tau may contribute to the clinical trial results. Our model is translational, so it can describe A-beta or tau pathology in different types of preclinical species. Parameters of the model different between mouse and humans can be scaled using standard allometric scaling, but regarding specific parameters for amyloid beta or tau, we used additional calibration based on the data for specific mechanisms. For example, it could be mutation or overexpression that could lead to increased production of A-beta or tau or increased aggregation rate. So we were able to describe A-beta mutations in the mouse, tau mutations, or 3X transgenic mouse, which carries both type of mutation. To describe difference between healthy and AD subjects, we used our disease driver model that have been mentioned before. Preclinical mouse models were used to validate our understanding of mechanism and pathophysiology on the different preclinical data for treatments. And after such validation, we tried to perform simulations for different types of treatments in Alzheimer's disease subjects. Now let's consider what our model demonstrated in comparison with the lately shared aducanumab clinical trial data. This antibody in our model can prevent secondary seeding of new oligomers of amyloid beta because it can bind to the surface of fibrils. And in addition, in vivo, this antibody activates clearance of amyloid plaques by microglial cells. Previously, we have satisfactorily described reduction of soluble and insoluble forms of amyloid on our model of transgenic 2576 mouse. And now we tested our model and compared with the data for aducanumab in human. Here you can see how our model describes reduction of amyloid deposition as measured by positron emission tomography by SUVR at the week 78. We have described two treatment arms. In the low dosing treatment arms, patients were titrated up to 6 milligram per kilogram, and in the high dose treatment arm up to 10 milligram per kilogram dosage. And further, we will refer them as a low and high dosing level. Our model satisfactorily describes reduction, but it underestimates difference between the dosages, which can be seen here as shown by the green color. We have also validated our model on the data available for tau pathology. Our model describes more or less satisfactorily a reduction of tau PET as compared with the medial temporal composite data and approximately, just like with the data, there is no significant difference between low and high dosing for reduction of tau deposition. Then we compared with the data our model predictions for CSF data on phosphorylated tau on the difference versus baseline. And here we see that actually our model underestimates effect of antibody on phosphorylated tau as well as dose dependence. We may assume here that actually in our model, sensitivity of tau kinase to A-beta and to A-beta oligomers can be underestimated. And here, although the model was taken into account different in vitro data on the influence of A-beta oligomers and tau, we see that there was not enough information about interaction of A-beta and tau phosphorylation. To be able to capture difference between low and high dosing, we also have screened different variables in our model, variables related to tau or A-beta pathology or neuronal homeostasis. And regarding difference between this low and high dosing regimen, there was no significant difference for most of model variables. So, for example, we can see here that there is absolutely no difference for total quantity of A-beta 42 oligomers, and moreover, the effect of therapy on the total quantity of these oligomers is very modest. But if we consider concentration of A-beta 42 oligomers free from antibody binding, we will see significant difference with a much higher reduction for high dosing levels. So first of all, we can see that concentration of antibody actually determines the availability of free oligomers. And probably if there is some difference between dosages in toxic effects, then it probably should be exerted, toxicity should be exerted by free A-beta 42 oligomers. So to test some of our assumptions, we also wanted to perform some tricks. We wanted to validate our model on established correlations known from the literature. For example, it is known that cognition and cognitive scores correlate with hippocampal volume. And it is also hypothesized and discussed a lot in literature extensively that, of course, K-toxicity is exerted by A-beta 42 oligomers. So let's now assume in our model that hippocampal volume explicitly depends on concentration of A-beta oligomers, and then calibrate the parameter of this dependence on hippocampal volume annual percentile change data available from the literature. So, we can try to answer two questions. What effect on hippocampal volume in the model will be predicted in aducanumab trial simulation? And would measured cognition scores really correlate with model predicted hippocampal volume change? So indeed, we see that if we consider hippocampal volume to be dependent on A-beta 42 oligomers, then there will be some difference in hippocampal volume change between low and high dosage, with a much higher decrease of hippocampal volume for placebo group. And if we align predicted hippocampal volume change with the data taken for both treatment arms and for both trials of aducanumab, ENGAGE and EMERGE, then we actually see a very good correlation between delta ADCS, ADL, MCI, and delta hippocampal volume. Of course, this is just a trick that can be performed by the model, but it shows us that probably additional brain volumetric data, especially for hippocampal, would help in further model validation, but unfortunately they were not available from the shared data. And this illustrates how a model containing different types of variables can be used to explore different relationships between measured data and predicted concentrations. And at the final part of my presentation, I'm going to demonstrate what our platform would predict for combinatorial treatment. Now, when, for example, aducanumab is approved, we ask a question, how can we boost its efficacy to obtain much better clinical results? As our model contains different types of intracellular processes, we can try some treatment targeting these intracellular processes. For example, autophagic system, which is critical for the protein degradation. Previously, when we were developing our neuron homeostasis model, we verified it on the in vitro data for rapamycin treatment. And that's how we verified some of the parameters of rapamycin effect on autophagy. And then we have taken preclinical data for transgenic mouse to validate our model independently. And model satisfactorily predicted a reduction of insoluble tau by rapamycin treatment for 2 dosing schedules that were applied at that data set. After this validation, we performed simulations for human separately for rapamycin or for rapamycin in combination with aducanumab and compared with results for aducanumab high dosing treatment. Results for PET-SUVR for A-beta and tau demonstrate here that indeed, actually rapamycin demonstrates already some efficacy for decrease of tau deposition, and this efficacy is much higher than for aducanumab. And a combination of aducanumab with rapamycin would allow even better reduction for tau, for example, in cortex. Regarding other potentially toxic species such as tau oligomers in hippocampus or free A-beta-42 oligomers, again, efficacy is much higher for rapamycin than for aducanumab alone, and moreover, rapamycin may contribute to the reduction of free A-beta-42 oligomers, although aducanumab is quite effective in reducing them. And finally, if we consider hippocampal volume, again, combination of rapamycin with aducanumab allows us to improve results and allow for better result on the reduction of hippocampal volume. So finally, the key take-home message from me is that QSP modeling is a valuable tool, first of all, for the exploration of different disease mechanisms and disease drivers. And simulations performed by quantitative systems pharmacology model allow for establishment and checking of existing biological hypotheses. And finally, QSP model allows for combining data from in vitro, from preclinical studies with the model provided description, and it allows simulation and testing different treatment schedules and combinations. Thank you very much.